So in previous videos, we presented the alternative pathway of complement activation. So if you recall, the complement system can be activated by three different mechanisms. And we talked about the alternative pathway activating by C3 interacting with the surface of a pathogen um, and leading to complement fixation, pathogens being covered in covalently in C3b proteins, and that having an effect on the immune response to help get rid of the pathogen. So in this video, we're going to talk about the classical pathway of complement activation. So it's another way for complement to become activated and for complement fixation, which is again C3b, is to be affixed covalently to the surface of a pathogen. So the reason that we are mentioning it now is because we introduced in previous videos the concept of cytokines such as IL-6. So we referred to the fact that macrophages have toll-like receptors on their surface. They can recognize pathogen molecules, and when they do, the macrophages will signal by producing cytokines that can act locally or systemically. Here we're talking about interleukin 6, IL-6, being produced, entering the circulatory system, traveling to the liver, and the liver producing a protein called C-reactive protein, CRP. Now, what we're going to see is that CRP is going to get released into the circulatory system, and it can travel throughout the body. It can be in the circulatory system in the blood. It can enter inflamed tissues because of increased vascular permeability, right? It's a protein. And CRP is going to be used as another mechanism to recognize pathogens. So another recognition mechanism. So again, what CRP made by the liver, what's it going to do? Well, physically, what CRP does, C-reactive protein, is it binds a molecule called phosphorylcholine. All right, well, what's phosphorylcholine? It is a molecule found on the surface of some pathogens in bacterial cell walls, in the cell membranes of some fungi, on the surface of some parasites. So if a pathogen has phosphorylcholine on its surface, then CRP will bind to phosphorylcholine. So it's a recognition mechanism. And this is going to trigger the classical pathway of complement activation, which we'll see on the next slide. So right now, so right, all the CRP is doing is binding phosphorylcholine. Okay, great. So here's a pathogen, and it's got phosphorylcholine, and we have had an inflammatory response. We've got IL-6 going to the liver. We got the liver making CRP, and now our CRP has gone throughout the body and found some pathogens covered in phosphorylcholine. So CRP is going to bind. Okay, well, what's that going to do? Well, we have to introduce a new complement protein. Actually, it's a complex of complement proteins called the C1 molecule. So C1, so we've learned a lot of complement proteins, right? We had C3, we had C4, we had C5. Wait, we didn't have C4, but we will shortly. We had factor B, factor D. Here's C1. The C1 molecule is actually made of a bunch of different complement proteins. So there's a protein called C1q, that's a complement protein, and in this drawing, the C1 proteins are the C1q proteins are the sort of those long stalks, and they actually have the physical interaction with the CRP protein. So this large sort of broom-like complex is made of mostly C1qs. And the C1q protein in the C1 complex actually binds to the CRP protein. Interesting. All right, so within this C1 complex, the C1 molecule, there are two other proteins, two other complement proteins called C1r and C1s. And these proteins are enzymes. What kind of enzymes? Proteases. But remember, proteases can be active or they can be inactive. So when the C1 molecule is produced, and remember, complement's present all the time, made um, constitutively in your circulatory system, right, by your liver. So you make C1 molecules, the C1R, C1S, C1Q assembles into this complex. Those proteases, not active until C1, the complex, binds CRP. When C1, specifically the C1Q, binds, triggers a change in the C1R protease and activates it. So now we have an active protease. And what C1R is going to do, it'll first cleave another with C1R. Remember, there are two C1Rs in this complex. And that C1R will then cleave the C1Ss. And the C1S, also a protease. So we're activating proteases. We got a little protease chain reaction here. Okay, C1R, C1S, proteases. Okay, all right. Proteases cleave proteins. They cleave substrates, right? So the substrate of C1R, C1 is itself, and then C1S. The substrate of C1S is, or I should say are, two other complement proteins called C2 and C4. So we're introducing some new complement proteins here. So C1S is a protease, and once it's active, and again, it's active because of this little chain reaction, C1S will cleave C2 into C2A and C2B. It'll cleave C4, or C1S will cleave C4 into C4A and C4B. Okay, well, that's interesting. What do these proteins do when they're cleaved? Okay, well, let's see. First, let's talk about C4. So when C4 gets cleaved, C4B, it looks and acts a little like C3b. Remember when C3 got cleaved into C3A and C3b? There was a very reactive thiol ester bond in C3b, which allowed it to interact with molecules covalently, and it got affixed to the surface of pathogens. Well, guess what? C4b has a thiol ester bond. And when C4 is cleaved to C4A and C4b, that thiol ester bond is exposed, and C4b can covalently attach to other things like the surface of pathogens. All right, so now we have attached another complement protein covalently to the surface of pathogens. So this is another complement fixation. We're fixing proteins on the surface of pathogens, marking them, which is a good thing. So when pathogens are marked with C4b, what does that do? Well, let's bring in C2A. So don't worry about C2b. We're just talking about C2A right now. C2A binds C4b. This produces an enzyme we're going to call the classical C3 convertase. Now, you've heard the term C3 convertase previously, right? C3 convertase, there was a soluble C3 convertase, there was an alternative C3 convertase, now there's a classical C3 convertase. All of them share the same function. They are going to convert C3 into C3A and C3B. This is a protease, right? The classical C3 convertase is a protease made of C4B and C2A, and that will take C3 molecules, which again, C3 proteins in the bloodstream, in your fluids, in your humors. It's going to cleave C3 into C3A and C3B. Right? And what's that going to do? Right? We know C3b, very active thiol ester bond, boom, start fixing to the surface of the pathogen. So we are marking the pathogen with C3bs. And we know what C3bs do, right? When they're fixed to the surface of the pathogen, they activate the effector mechanisms of complement, like opsonization. A macrophage should come in here and bind the C3bs with the CR1 receptor. Macrophage eats the pathogen, opsonization. C3A, we recall, is an anaphytoxin. It can induce inflammatory responses. So we got inflammation going on here. I will mention that C4A, right, I sort of skipped over it when C4 got cleaved in C4A and C4B. C4A, another anaphytoxin. Not as powerful as C3A or C5A, but it's an anaphytoxin. Okay. Can we make the membrane attack complex? Right? Doesn't that help get rid of pathogens? It does. So if you recall, there was the alternative C5 convertase we made in the alternative pathway. Well, I'm here to tell you that we're going to make a C5 convertase in the classical pathway by joining a C3b to the classical C3 convertase. So again, we've got C4s and C2s being cleaved by the C1s. And so as that's cleaved, we're forming C3 convertases, the classical. We're also forming C5 convertases. So the classical C5 convertase is when the classical C3 convertase, C4b, C2a, gets joined by a C3b. So this tri-complex actually now functions as a C5 convertase. And you recall what the function of a C5 convertase is. It cleaves C5A, it cleaves C5 into C5A and C5B. And what's that third effector function of complement activation? Right, C3b will begin the formation of the membrane attack complex, joining with C6, C7, C8, a bunch of C9s, embed into the plasma membrane of the pathogen, poke a hole in it, membrane attack complex, and lyse it. And then C5a, we know, anaphytoxin, help induce inflammation. So this is the activation of complement via the classical pathway. So again, classical pathway was triggered by the production of CRP from the liver, which, if it binds to phosphorylcholines on pathogens, will start this chain reaction using the C1 molecule, leading to the formation of the classical C3 convertase, which can lead to the formation of the classical C5 convertase.